Okay, cool. So my name is Weston Jackson. I'm a recent graduate from Columbia University, Department of Computer Science. I did this work with Ipsita Agarwal and Professor Itzik Peer on modeling microbiome samples. And I now work at AppNexus in their optimization department. So before we get into what the algorithm is and what the results were, I just want to define what exactly a microbiome is. And a microbiome constitutes microorganisms in a particular environment. And in our case, we focus on the body. The Human Microbiome Project is a project that's dedicated to analyzing microbiome samples in order to better understand human health and disease. Normally, microbiome analysis is done by taking some samples of a microbiome from a person, using some protocol to make high-dimensional vectors, projecting this down into low-dimensional space, and then running a clustering algorithm so you get a better understanding of how these microbiomes are clustered and how to understand the bacterial samples. In our case, we tackle microbiome clustering while allowing samples to belong to two clusters. So our data consists of 1,500 mid-vaginal samples from the NCBI database. And these were processed using a standard protocol for microbial analysis to create high-dimensional vectors. And then we performed a dimension reduction plot, which revealed 5 clusters. And note that each point is one sample in this graph. So there are a few problems. We had 5 clusters, but there's a lot of samples we noticed that lie in between microbiome, or in between the clusters, basically. And it's ambiguous where these samples belong. In addition, we could say that these samples maybe belong to the closest cluster, but this could change where our cluster representatives end up, and it could shift them toward these in-between points and toward outliers. In addition, this sort of pigeonholing doesn't actually reflect the fact that samples often reflect a mixture of two populations. So we kind of asked ourselves, is there perhaps a better way of understanding and describing this data? Our idea was to allow samples to have two-way assignment. And formally, we say that each sample xi belongs to some two-way cluster, Cj, J prime. And we added an extra parameter u, which determined the proportional distance each point lay in between its two representatives. So as an example, these are three cluster representatives, C1, C2, C3, and then we would get all these two-way clusters. So for example, C12 is between C1 and C2, but closer to C1. C21 would be closer to C2, and between C2 and C1. So we have these sort of two-way clusters as a way of identifying what's going on. So now our issue is, how do we learn this extra parameter u? So we used a variant of kMeans. And kMeans, if you're familiar with it, k is the number of representatives, and it learns k representatives to minimize the following objective functions. So the squared distance of each point to its closest representative. Now there's a generalized version of kMeans where each data point has a vector of representatives, or a coefficient vector. And this coefficient vector, each point has one coefficient for each representative. And this generalized version of kMeans is the following. And now in this case, this matrix C is the matrix of all the representatives, and each phi I, there's one coefficient vector for each data point. So now two-way kMeans is this exact same generalized kMeans, but we have added constraints. First of all, our coefficient vector only has 2 non-zero coefficients, and this is because each point can only belong to 2 clusters. So our coefficient vector would look something like this. One representative would have a coefficient ui, and one would have 1 minus ui, and these also sum to 1. So in particular, we have a generalized kMeans objective, but we have constraints on the coefficient vector to ensure that each point, we're looking at it from this perspective of each point belonging to 2 representatives and having coefficients that sum to 1, because these are supposed to be proportional, such as like 0.25, 0.75. Now we have to, now it comes we need to focus on learning what this coefficient vector is. Now, in generalized kMeans, this is done by alternating minimization with respect to phi and c. So alternating minimization with respect to the coefficient vectors and then with respect to the representatives. So we're going to do something that's very similar, but because we have these added constraints, we had to make a few changes. Specifically, minimizing with respect to phi, we can't, because of the added constraints, we substituted in the fact that our coefficient vector phi I only has 2 non-zero entries. So we substituted this into the kMeans objective, and then we fixed these 2 representatives. And then if we do that, we can find what UI is, or the kind of proportion parameter, by just taking the derivative, and there's only one critical point. Now the issue is we just fixed these 2 representatives. We don't know what they are. So the best way, in our case, we consider the fact that the number of representatives is usually a constant, very small, much smaller than the number of points. So looking at doing a grid search over all possible combinations actually doesn't really affect the time complexity too much. So in this case, we just did a grid search over all combinations of 2 representatives and then maximized or minimized with respect to each one, and just found the value of UI that minimizes the objective the most. Now, minimizing with respect to C is actually no different from generalized kMeans. You can actually use ordinary least squares to find the optimal matrix of representatives. I can go into more details about this, but basically it's exactly the same. It's just ordinary least squares. You get the following objective, and you can find CI transpose. It doesn't really have a good kind of intuitive interpretation, but in terms of analysis, it's pretty straightforward, and you can do the entire thing with just one big matrix multiplication. And then we alternate, minimizing with respect to phi and minimizing with respect to c until we converge. So that was a lot of math. Now, what actually happens? So this was the plot we saw before. It was just an example plot. The diamonds represented the actual representatives, and this was a plot I simulated of points belonging to 2 representatives with a certain proportion parameter. The triangles is the learned representatives by kMeans. So kMeans kind of skews the clusters toward the center to compensate for the fact that many points lie in between clusters. So two-way kMeans, by learning these sort of lines, actually gets very close to where the cluster representatives actually lie. So now we can look at the real data. The same samples as before. Our algorithm converged after 5 rounds of two-way kMeans, and in our case, we initialized with kMeans, which actually helps because you get close to where the clusters are, and then you just kind of learn and kind of exactly where the lines kind of match up. One potential issue and opportunity for further research is the fact that we noticed actually that there's actually some nonlinearity between the clusters, which does affect where our representatives end up. However, I can answer more questions about that, but for our cases, it actually didn't really make a difference, or it didn't matter too much. So this is the comparison before and after. The darker points are part of the clusters. The lighter ones are like in between 2 clusters, more or less. So now that we have these clusters, we can ask ourselves a few questions. One, what's the most abundant species per cluster? And we can ask this because each sample has a most abundant bacteria in it. So what species are most common in each cluster? We notice that most clusters have 1 dominant species. So C1 through C5 are the 5 clusters. And notice that C1 has 1 dominant species, C2 has another dominant species, and C4 and C5 have other different species that are the most dominant in that cluster. And C3 is kind of a diverse cluster. We really don't know much about it at this point. But if we ask, what's the most abundant species per cluster pair, we can learn more. Basically, which bacteria are most common in each two-way cluster. We're going to use the same notation as before, and just as a refresher, if that cluster C1 and that one C2, these points would be lying in C1,2, and these points would be lying in C2,1. So that we get this big table. And let's just make sense of this. These are all the points that are closest to cluster 1, and they have the same most common species of bacteria as before. These are the points that lie closest to cluster 2, the same most common bacteria. Same for C4 and C5. This is sort of unsurprising. But for C3, we noticed that most of the bacteria actually lied in these subclusters, C3.1 and C3.4. So between clusters 3 and 1 and between clusters 3 and 4. So our results are that C3.1 and C3.4 are very different from each other in terms of how they're made up compositionally. They're also very different from the other clusters. So in this way, perhaps C3 should not be seen as a sort of discrete cluster, but maybe consisting of 2 subclusters. And then in that way, perhaps there's really 6 clusters. In addition, there's also, with this new proportion parameter U, it's actually, it's kind of intuitive to this sort of analysis, because a lot of times the microbiome can change. For example, if someone has a disease or is unhealthy, the state can change. And in that way, the proportion parameter U can kind of, it can be seen as like how people are changing from one state to another state. Anyway, in conclusion, as microbiome data becomes more and more available, standard methodologies see clusters as being discrete. And samples often reflect a mixture of 2 populations, and because we allow samples to be weighted averages, we think that two-way kMeans allows us to better understand the microbiome and better understand the human. So that's it.
Thank you, Weston. References.